Good afternoon and a very warm welcome to this Genetic Therapies and New Modalities panel, co-chaired by Michael Rice from Cello Health Consulting and myself. My name is Strohmenger from Wellington Partners. We have as panelists Heinz Lubin from VAXIMM, Jay Olson from Oppenheimer, Regan Jarvis from Anocca, and Sondra van Deventer from Forbion. So a very competent panel, and I think it will be a very exciting discussion. And maybe we start with a short introduction round. And just briefly on myself, I've been in venture capital for 23 years, always at Wellington Partners. One of the managing partners based in Munich. We invest all over Europe, and my personal background is in medicine and economics, and about half of what we do is in therapeutics. The other half is in medical devices, diagnostics, and healthcare IT. So, Michael.
Thanks, Rainer. I'm Mike Rice, Vice President at Cello Health BioConsulting. I lead the advanced therapeutics practice that we created about 5 years ago, just as we started to see this field take off, largely with the advent of the initial CAR T products as well as AAV therapeutics. But since then, we've really kind of broadened and worked with hundreds of innovators in both the oncology and non-malignant disease side. Largely what we're doing is helping them harness the potential of these broad-based technology platforms and help them with decisions and how do you decide how to deploy capital onto, lead development programs, what indications to go after, how do you invest in manufacturing capabilities, how do you gather the right talent, and all the things that you need to do to draw more capital for your sort of next steps in the development of the program. For pharma, we traditionally been doing futurescaping, looking at what might gene and cell therapies do in the future that might impact their businesses. However, now it's become more of how do we balance our portfolio with the right assets and platforms that were competitive with our other pharma competitors, and how do we operationalize that and make a business out of it? So that focus has really quite changed. But it's great to be able to sort of fertilize learnings from the buyers as well as the innovators. So look forward to the discussion today. I will hand it off to the panelists for their self-introductions. So why don't we start with Heinz, since you're up here in my top left.
Okay, thank you, Mike. My name is Heinz Lubin. I'm the CEO and co-founder of VAXIMM. I'm a pharmacist by training. I started my career in the pharmaceutical industry 26 years ago in Paris and in Munich when I did learned clinical research in Servier. And 18 years ago, I stepped into biotech as I joined one of the first biosimilar companies in Germany, the one of the Ratiofarm group, which is now part of Teva. I developed the first biosimilar GCSF into the market, up to market launch, and the second generation BioBetter, the new Lasta BioBetter, into clinical phase 2 from project implementation. And now I'm a CEO and co-founder of VAXIMM. We are developing an oral DNA vaccination platform consisting of two components: the live bacterium, which enables the oral administration, and the eukaryotic expression plasmid, so the DNA, where we can encode whatever target antigen we want to encode. We can encode full-length proteins, we can encode short peptides, a very flexible platform, and we have an off-the-shelf product, which is our, let's say, lead compound, which we develop as a clinical proof of concept in recurrent glioblastoma. And we have the personalized neoantigen approach, which we want to bring into the clinic within the next months, and we want to treat the first patient in summer, do a small signal finding study, and go extend the study from there. So thank you for
being on this panel. Thank you, Heinz. So Jay, why don't you go next?
Great, thank you. Hi, I'm Jay Olson. I'm a biotech analyst at Oppenheimer in New York. I started my career at Pfizer doing mostly business development and marketing for 16 years. And for the past 12 years, I've been doing sell-side equity research on publicly traded companies, mostly small to mid and large cap biotech companies in a number of different therapeutic areas. And it's a real pleasure to join your panel here today. Our investment thesis is really based on innovation, and we look for potentially disruptive therapeutics in areas of high unmet medical need. And some of the technologies that we're excited about include antibody drug conjugates, bispecific antibodies, and genetic medicines. And I'm looking forward to the discussion. So thank you for inviting me.
Thanks, Jay. Regan, why don't you go next?
Thanks, Mike. So Regan Jarvis from Anocca here in Sweden. I'm by far the least experienced on this panel, as usual in these forums. My background in industry is at Anocca. I'm a basic scientist. Studied biochemistry in my native New Zealand. Long story, but ended up here in Sweden, starting up Anocca as a scientific founder. And we are a T-cell biology company. So we've built a fairly modular platform, which is based on standardized, reproducible cell-based assays for T-cell biology. And today we're focusing on TCR-T therapies in the oncology space. We have a pretty deep pipeline of TCR assets, which span a range of target types and applicable to a range of hematological and solid tumor indications. We've also built out an in-house manufacturing platform, and so we can independently deliver some of these early programs, the TCR-T programs, to clinic. And they'll be coming through to first in man, all going well, second half of next year. So exciting times at Anocca, and happy to be here and looking forward to contribute to the discussion.
Thanks, Regan. Sondra, yeah, thanks.
Yeah, Sondra van Deventer. I'm trained as an internist. Actually, still am a professor of medicine at the University of Leiden. And I was one of the founders of Forbion and currently an operating partner. And at Forbion, we started and invested in many gene therapy companies, including Bluebird Bio, BioVax, Replimune, Hookipa. And I'm also a founder of Unicure, have been the CEO and CMO. And until last year, I was the EVP of research and product development. So brought, I think, about 6 programs into the clinic. The most recent were the hemophilia B program and the Huntington program, which was a gene silencing program. So currently operating partner at Forbion and CSO of a Chinese company, Porton Bio, and I'm building a very large AAV manufacturing facility in Suzhou as we speak.
Great, thank you. Okay, well, let's start the discussion. And the topic of this panel, genetic medicines, new modalities, is really an area that's been at the front of investor interest. As Jay had mentioned, and a couple of you had mentioned through this. And it has been for a couple of years, and this year was kind of a year of uncertainty when everything went virtual. How were things going to progress? How were roadshows going to be done and everything? But what we experienced was a really rapid pace of Series A's. Some of our clients did Series A's, crossover rounds, and right into IPOs, and all in several months over the summer. And we see this going on in the whole field, and it's driving the rapid pace of innovation in advanced therapeutics, really broadly, with a lot of different dissimilar technologies that or may be going to very unique patient populations, and many of them are chasing the same patient populations and trying to prove a level of differentiation, or at least prove the platform where they may take to a unique patient population as a next step in their development plan. And so, I guess, the interesting thing is just the level of investment and the fact that this is all going on in, largely pre-POCs. Even the pharma partnerships have been buying in early before proof of concept at high valuations. And the question I have for the panel, maybe we'd start, is why is this area so interesting, and why are everybody sort of jumping in right now, both from the investor side as well as strategic partners and everything. So I think we'll start the round of questions with the investors just to set the context of, how they evaluate these, early pre-POC programs and make investment decisions around them. So who wants to kick it off? Maybe Rainer, why don't you kick us off in this part of it?
Sure, yeah, happy to. So of course, new modalities, new technologies create new opportunities to bring novel therapies to patients. That's obvious, and I think what we see is the result of a lot of scientific progress that has been achieved the last 2 decades, and that now allows for creating much more targeted therapies and create products that simply go beyond what was possible with classical pharmaceutical approaches before. And I think for me, it's kind of a balance between, of course, each therapeutic modality has certain strengths that you need to understand, and you need to pick the right indications. You need to find the fastest way through clinical development, and really find your niche in unmet medical needs. What we see today is sometimes that there are many early stage platforms that do not necessarily follow that route. And currently, there is a lot of capital available, especially in the US. I'm wondering, will that also lead to some kind of disappointment in a number of cases? And are we now with some modalities in the first wave or in the second wave, what we have seen with so many technologies, that initially there's a lot of enthusiasm. There is already a first wave of successful IPOs, successful trade sales. But then until you have a product on the market that is really successful, it may take another 10 years or even longer. And maybe, Sondra, you can share your view, because you have been part of that a couple of times.
Sure. Yes. I agree with you, Rainer. So, but the direct answer to the question, I think, is that we can do things that we couldn't do before. And some of them are really paradigm shifting. So, for example, if you take the How B Uniture program for hemophilia B, there's patients out there that have now expression for 4 or 5 years continuously. So there's no decline. And that's a 30-minute outpatient infusion. Now compare that with replacement therapy, where you have to have, once weekly or twice weekly infusions. And the other thing is that patients on gene therapy simply don't bleed. Whereas with replacement therapy, there are still a lot of intercurrent bleeds and still a lot of invalidating complications, such as joint bleeds and what have you. So there is a paradigm shift that's ongoing already, for example, in the hemophilia B field. And now, obviously, that is sort of low-hanging fruit, I would say, because this is a disease that you really can cure if you have sufficient expression. And when we started, we had like 2% or something like that expression, very much on the low end. But now patients have long-term 40 to 50% expression. That's really, I would say, almost curative. So then the next step, I think, if you think about it, there are so many neurodegenerative diseases for which there is no real treatment right now, only supportive treatment. And these are diseases that I would say would be, next obvious target. So, for example, our Huntington program is a good example, but that's just one of the many neurodegenerative diseases where these technologies offer really new potential paradigm shifts in the treatment. So I think, the enthusiasm, I would say, should come from investors because of this. And then I also agree with Rainer, there's an initial hype and then there's a long term of, disappointment with gene therapy that lasted maybe even something like 20 years. But I would say there's now clear examples where we have things that work. And a similar, shift or a similar kinetic, I think I would expect would happen with gene editing. So that's how it goes. But there are definitely paradigm shifting therapies that have come out of gene therapy now. I'll leave it here.
Yeah, and okay, Jay.
Oh no, I was just going to say I agree with both Rainer and Sander. Scientific progress has driven a lot of excitement amongst investors as the rate of innovation has really accelerated. I think the COVID pandemic has also demonstrated to the world that difficult medical problems can be solved quickly with novel and effective new vaccines and therapeutics. And biotech investors in genetic medicine and other advanced therapeutics have been rewarded with some of the most attractive investment returns that we've seen over the past year. Since I focus mostly on public companies, most of the investors that I work with prefer clinical data, but we are seeing more and more companies going public pre-proof of concept, companies with well-validated technology platforms. They often have partnerships with academia or corporate partners that really provide external validation to their technology. So as a public stock analyst, pre-proof of concept companies are difficult for me to value, but they are becoming increasingly more common, and they're also delivering, I would say, outsized returns for investors in some cases.
Mm-hmm. So we brought up, a number of different advanced therapeutics platforms from, AAV therapeutics, CAR-Ts, adoptive cellular therapies, gene editing, mRNA vaccines, just in this little short, part of our discussion. And, and some of these areas are broadly looked as being, hot investment areas. Even though they're group buckets of, very different approaches in some cases. If you take the gene editing space, from, tools for just doing gene augmentation down to, base editing or epigenetic modification, they're vastly different approaches, but they all get sort of lumped in, for many investors that may not be as sophisticated as, some of the panelists here today. How do you sort of evaluate, especially this pre-POC, Jay, you mentioned you like clinical data and see that in public companies. But how do you evaluate dissimilar technologies and decide if it's going to be an investment? And what sort of advice could you give the biotechs of how do they position themselves for the the hot bucket to be in versus, the cold bucket that, investors are saying is not really ready for prime time? Who wants to start that?
Well, one of the features of this business is that you really need to, probably have access or have developed a platform that you can work on. So, for example, if you take viral gene therapies, then immunogenicity is a big one, and of course manufacturing. So, I think companies that cannot offer the possibility for a partner, large pharma partner, to manufacture their product at a larger scale, definitely are not as attractive as those who can. And that's not a small thing because there have been enormous acquisitions of technologies that were quite interesting but didn't come with that manufacturing platform, and large pharma has found themselves actually in a position where they really couldn't even do it themselves because these are not technologies that large pharma really masters. So some of the very costly acquisitions that have been done by pharma now were actually solely for manufacturing. So that's an aspect that I think is underestimated.
Yeah.
And then the other thing is that if we talk about gene therapies now, it's not simply gene expression, but it comes, there's gene silencing, there's vectorized antibodies, there's a whole list of other things that you can do with gene therapy. So we're looking at gene therapies basically taking over part of the protein replacement market, part of the silencing market, what you now do with silencing RNAs, and potentially part of the antibody market, because you can do vectorized antibodies. So that's basically something you can look at, and that's what pharma is actually looking at.
Yeah, absolutely. Yeah, and I did pop into the panel before this, and they were also talking about investors are highly interested in platform technologies rather than single asset deals and stuff like that, and but your ability to maintain and raise additional capitals is really dependent on how successful you are with the first programs that you move into the clinic, right? So how much of it, or do you, how much of your diligence do you put on that lead program, versus the overall platform, and how does that, affect your investment in the opportunity?
Well, if you look at Series A now, rounds for these sort of technologies, they're very often in the realm of $100 million. And why is that? Because investors realize that you need to finance these companies very well, that you don't want to be stuck in a sort of, valley of death, when something fails and you don't have the capabilities of salvaging the program. But I think, investors also really look at teams now. Is this a team that can develop this? And if something goes wrong, get it on the right track again. So I think investors really look at track records. They look at capabilities. They look at things like the possibility to manufacture products, and that's basically how it's being done now. It's very uncommon to see like a 10 million Series A for such a company now, and the reason is that it's highly risky to do that. It's a very risky bet to just put 10 million there, maybe with a team who's never done this before, maybe without an outlook for how to manufacture this. So that's why you see these enormous rounds now. It's not only risk; it's actually de-risking.
Great. Jay, you want to comment maybe on the consequence of the lead indication and what you see when they start to actually produce clinical data and the impact?
Yeah, I mean, I agree with Sander that the platform is important. We tend to focus on the lead program and start to try to identify the commercial potential for the initial indications that will be pursued. But again, understanding the platform, I think, is where you're going to show differentiation from competitors early on, the ability to adapt to multiple targets and diseases, and bring in new candidates in case the lead program fails. But what we like to see in the lead program is compelling biology, predictive animal models. We also want to see pursuit of areas of high unmet medical need with clear regulatory pathways. We look for something that's potentially disruptive, offering a significant efficacy advantage over the standard of care, and potentially first-in-class or best-in-class approaches, in some cases to previously untreatable diseases where there may be no other therapeutic options. And we always look for disease modification, not just a palliative treatment, but something that's potentially curative. And then there's always the question of chronic versus the once and done treatment. That's another debate. But I think pricing and reimbursement for once and done treatments are becoming more widely acceptable with increased understanding from payers. And obviously an economic argument must be constructed. So we look for something there as well. So those are the kind of things that we look for in the LEAD program.
Great, thanks. Rainer, do you want to add anything to this before we go on to the biotechs?
Yeah, I think, I would really want now to hear the view of Heinz and Regan, how they address these issues and position themselves in their fields in order to make them as attractive for investors as possible and really to attract these huge amounts of capital that are required to successfully compete with their peers, especially in a field, for example, like immuno-oncology, where you have so many different approaches, and it's almost impossible as an investor, to have the full overview. I don't know if anyone can have that. Heinz, how do you approach that?
Yeah, so thank you. Yeah, what we are developing, as I said, is the oral DNA vaccination platform, which has some, let's say, key features which I think are quite unique. On the one hand, of course, it's the oral administration, which is an advantage to patients. Patients like it, doesn't hurt, there's no injection site reaction, but it's just one feature of the platform. On the other hand, I'm hearing about, let's say, manufacturing capabilities. So for us, we have a very easy, very straightforward manufacturing with reasonable cost of goods. You can imagine that the bacterium which carries the plasmid is the product. So it's just a fermentation, and the plasmid replicates itself inside the bacterium when we do the fermentation. So that is how the system has been set up. It's a very fast manufacturing. It's just 10 to 12 hours. So you can do it overnight, for example, and that's a really, let's say, true advantage with regard to the personalized neoantigen approach, together with the low cost of goods. And what we can also do in comparison maybe to viral carriers, which are very popular at the moment as well, is we can do homologous boosting. So we have done an initial vaccination plus 4 weekly or monthly boostings in some patients now for 4 years, which survived as recurrent glioblastoma patients. And so we see that these are some advantages. And we want, of course, tick all the boxes relevant for the investors. I'm coming back to what you said in the let's say in the last minutes. And on the one hand, we need very good scientific data. On the other hand, we need some external validation. And I'm not only talking about scientific validation, also talk about validation, let's say, from regulatory agencies. For example, we did, with regard to manufacturing, with regard to preclinical development, the so-called ATMP certification by the EMA, which is some kind of pre-assessment for small biotechs. And we got the certification as one of the, let's say, rare companies. And now, as we go into personalized neoantigen approach, we saw for the first time that this is something which can be valuable to go ahead to get regulatory acceptance for another product, for a second follow-on product. We want to combine with checkpoint inhibitors, so that enabled us also with the personalized neoantigen approach to go directly into an approach where we can combine, and also in a first-in-human with an active dose. So we are trying to collect, let's say, tick boxes, also with regard to expert statements, with regard to environmental safety, as we are a GMO. And there we went to the Netherlands. We got a really extensive assessment, which takes 10 to 12 weeks at least. And now we have a public assessment report, which can be shown to potential investors if they have increased interest. So that's how we approach the different requirements from the investor side.
What is your killer application that you anticipate for your technology?
You mean in terms of indication?
Yes.
Yeah. So what we're
the
doing,
really low-hanging fruit.
yeah. So what we are doing, and I think that's also a matter of discussion always with the investors. We have chosen the indication of recurrent glioblastoma, which may not be the favorite indication for everybody, but we have a clear scientific rationale to go into that because the antigen which we target with our lead product is highly expressed. It's not only expressed on the tumor vasculature, it's also expressed on the tumor cells, and we have seen clinical responses with our cancer vaccine approach, with and without combining with checkpoint inhibitors. And we have seen anecdotal clinical responses, one complete response, one partial response in a previous trial. Now we are doing a trial together with Merck and Pfizer, where we combine with the avelumab, and we have seen again clinical responses, more than 50% tumor size reduction in the combination. And this is very reassuring. And what we want to do is we want to extend this patient set. We have now 12 patients, and 5 out of these 12 have experienced a clinical response. We want to extend it to another, let's say, 30. And then if we can confirm what we see, I think we are in a very good position to go ahead with regard to a regulatory strategy where we are currently working on, and with regard to FDA contacts in terms of, let's say, further assessment by the FDA for an accelerated approval in an indication where nothing works.
If I may, Heinz, can I ask you a question? Because I thought it was really interesting that you said, backbone responses to viral vectors if you re-administer. I mean, that clearly is an important point, in general, pertaining to re-administration. Yep. But there's also some sort of actuality to it, which is that, of course, some of our COVID vaccines are based on adeno backbones, and I just don't understand, why there's no discussion that, if there's a new mutant, and for some of these vaccines, some of them, like the Sputnik vaccine or whatever, they already do some serotype changing with the second boost. But if we have a new mutant in a couple of months or next year or whatever, how many times can we use adenoviral vaccines, right? And there's a clear difference here with some other vaccines like the BioNTech, Pfizer, and Moderna's. Maybe you can comment a little bit on that. Why platforms are really important.
Yeah, so I mean, we have seen with our bacterial vector at the beginning, we had the same, let's say, fear, and we were even by the agency where we were reminded, exclude patients who have been exposed to the prophylactic typhoid fever vaccine, which is your carrier. What we have seen in the first trial is that really the showcase patient, despite we had excluded patients which had been previously vaccinated, had the best response. So maybe if there is an IgA response, a pre-existing IgA response, the bacterium is marked, and anyway, the human body wants to get rid of this typhoid fever vaccine, so it's taken up into the Pias patches. Maybe that's even more efficient. We don't know, and so that's what we are trying to explore now in the let's say current trials and in the upcoming trials. Also together with the microbiome assessment. And I think that will be a very interesting data set, but you need more patients than just 10 or so.
Great. So Regan, so I'd love to hear about the TCRT platform a little bit more. And, Heinz is going in a very, challenging, high unmet need indication. You're somewhat, following Adaptimmune and, GSK and others with your lead program, I from what I understand, but you have a great depth of a pipeline behind that to go into more and more personalized, settings for different solid tumors and things like that. So I'd just love to hear a little bit more about, how you're going about creating value from your platform, in the position that you are.
Yeah, exactly. And just to start off, I mean, we've got a very different type of platform than, say, VAXIMM. So we're not about formulation delivery. We're about the analytics, target discovery, asset generation, and recognizing fully, you need to manufacture, you need to deliver these to clinic. And in the T-cell therapy space, that is, transducing or inserting your asset into an effector cell population. And we very much recognize that the field is moving very rapidly on that side. There's literally billions of investment going there, some big M&A activity in the pharma industry. And what we're doing is twofold. One, you said up front, basically, is we've got a deep pipeline, so it's decisions what to take into your lead programs, because all eyes are on that lead program, right? And we've selected AML, which is a high unmet need. It's a fairly big market. It's a tough indication. And the reason we selected that is severalfold. Not only is it sort of, it's a big indication, we're also using a target that has been pursued by Adaptive and GSK in the past, and that's NY-ESO-1. It's a really well-characterized target. And that in itself, de-risks our program because it's a validated target, essentially. And we've got some unique translational biology around the AML. But more importantly, the AML space allows us to do a relatively straightforward manufacture in the sense that we're doing post-hematopoietic stem cell transplant and doing a donor allogeneic manufacturing. This has been done before in an academic setting. It just allows us not to recover our autologous cell product from those very highly conditioned, very unwell patients, essentially, and rather take the HSCT donor as an allo source of the cell therapy product manufacturing input. And that's just a first step. We're looking forward to genuinely autologous programs. And a bit uniquely for a company the size of Anocca, we've actually invested in a manufacturing platform. And by that, I mean, it's the infrastructure, it's a facility, it's the know-how, the QMS system, so on and so forth. And being flexible enough in that infrastructure to incorporate both the donor allogeneic, autologous, and any off-the-shelf allogeneic platforms that we'd care to partner with or out-license to and commit our manufacturing capacity to. So, it's really a different type of platform. It's about target discovery, asset generation, and realizing, we need to progress things to clinic. There are lots of ways to progress TCR assets to clinic, and we're not going to marry ourselves to any one cell effector. It can be T cells, can be NKs, could be other immune cell types. We're not specialists in that area. We're specialists in the target discovery, asset generation, and building up a manufacturing platform and infrastructure that allows us to bring in different types of effector cells into which we can plug our assets. So it's a unusual approach in that respect, but we've got a good rationale behind both the indications, the asset selection, and maybe just to say is that we're not going after NY-ESO-1 with a single asset on a single HLA restriction. We're digging deep there and saying, it's not just A201, 50% of Caucasians. We're covering just in the disclosed programs that we've got running about 70% of the worldwide or major markets worldwide, let's say, through 5 different assets across 4 different HLA restrictions. So that's a differentiation in itself. We would say, of course, best-in-class assets, but a lot of these are first-in-class, and that people simply haven't made the TCRs against these HLA-restricted epitopes before.
Great. Any other panelists have questions for either Heinz or Regan?
Yeah, I think one question from my side. How do you overcome the problem that you know your technology best? You need to show the proof of concept, and if necessary, you need to show that in the clinic. And can you build a successful company without doing that on your own in case of doubt?
Yeah, I think that's a good question, Rainer. I was also thinking about what Regan just said. There is a little bit of a dynamic that very early companies come with a clinical indication, and then there's this sort of rat race for discounted cash flows or net present value because people want to calculate some sort of value associated with the program. I've had something like 60 PhD students, and they come up with an interesting experiment, and you say, well, this is really nice, But I say, it's really nice only if you show me that you can do this 10 times, okay? So there have been many, actually, companies that we looked at, and I said, forget about first indication. We'll just finance you to do just simple biodistribution or manufacture enough to do a monkey. That's sort of, a primate. So that's sort of basics. And that's, I think, where we need to be careful. we some programs have a high value and some companies have a high value, preclinically, but we really need to, force companies to do what Reagan is doing, just basically, make sure that the technology is working, that you can manufacture it, and then, indications will come. Whereas I very often now see very early companies with a bogus study about some disease I've never heard about, that they tell me they're going to sell their product for 600,000 euros or something like that per year, that sort of story, because that's the only way they can sell it. So I think that sort of dynamic we should be a little bit careful of.
Yeah, and I actually, the calculation I thought you were going to say was probably the calculation Jay was making was, how do we take a TCRT program, that provides proof of concept in a heme malignancy, AML, and then think about, what is the upside value in the 90% of solid, 90% of the oncology market is solid tumors. How do you convey the broader potential beyond, AML?
Yeah, sure. I mean, just reflecting back some of those comments, essentially, upfront, TCRT is an extremely immature field. I mean, what we're looking for are proof of concepts right here, not just on targets and indications as an entire field, quite frankly. And so AML is something we've chosen with the rationales that I mentioned. And at the end of the day, we don't see solid versus heme as being a T-cell problem. We see it as purely a target problem. And I think this comes from the CAR T space, where people are saying, yeah, CAR Ts are not effective in solid tumors. And I'd say simply, there are no good targets in solid tumors. That's why CAR Ts aren't effective there. That's simply not the case with TCRT. The T-cell receptors target HA-restricted epitopes you're seeing inside the intracellular sort of epitope space, the mutanome, whatever you want to call it. The targets there are absolutely rich. The challenge in the field now isn't how you're going to manufacture your cell product, how you're going to get it into the tumor microenvironment, whatever. It's really having a good way to identify targets and make high quality assets, and more importantly, define what a high quality asset actually is. What does a good TCR actually look like? And that's how we've been building out our technology platform and showing the value of it, is actually using our really precise cell-based tools to define what are safety benchmarks for TCRs, what could be efficacy benchmark for TCRs themselves as therapeutic molecules. So, it's yes, heme versus solid is important. A lot of the market potential is in solid, but we say that's targets, not T cells.
Yeah, and Jay, any comments on how investors look at heme versus solids, and off the shelf versus autologous? And
yeah, I mean, it's a great question. A lot of the advanced therapeutics that we're talking about and investors are excited about are obviously more validated in hematological malignancies. Solid tumors have been more challenging for a lot of technologies, not just CAR Ts, but bispecifics, antibody drug conjugates. So there are a lot of challenges there. But I would say that in terms of what we look for and what investors look for is really, in addition to the science and how it compares to competitors or other drugs in development, you might be going after the same target, like say Nectin-4, but can you do it better than anyone else? And can you do it in a way that will translate into superior clinical efficacy or safety or some other clinical differentiation? So I think those are some of the things that investors look for. And then can those benefits show superiority over the standard of care or other drugs in development?
Yeah, I think, investors, well, at least I can speak for myself as an investor, when it comes to, liquid or solid tumors, the early CAR T cell data were, of course, great. I mean, if you look at the long-term outcome of some of these trials are just stunning. I see a CAR T cell as a hand grenade with the pin removed, right? So it goes off, whatever you do, And so, it's easy if you're a B cell and that's the first cell you encounter, bang, there you go, right? But for solid tumors, there are so many more hurdles to overcome, such as going through the endothelium, then going into the tumor, and then taking care of not being checkpoint inhibited and all that sort of stuff. And so that if you have a hand grenade that goes off immediately, then it will never get there. So I think when we look at these sort of things, then at this point in time, we are generally more interested in T-cell receptor approaches because of exactly these issues. We're interested in some sort of proof that the cell actually gets in the tumor, so it's TIL-like cells. And then, of course, as we already briefly discussed, the whole antigen part, what is a suitable target or target antigen for this cell. So I think getting from just engineering T cells that immediately explode when they encounter a certain target. This is a much more complicated biological process to be solved, and there are more checkpoints there's a whole marriage also with antigen discovery and all that. And so I think we will get there, but it's definitely more complex.
Great. So I think we only have a couple minutes left. We do have an audience question here for Heinz. So I don't know, Heinz, if you can see it, but the question is, how does VAXIMM ensure that oral administration bacteria survive the low pH environment of the stomach?
Yeah, that's a very important point. So we need the live bacterium entering the small intestine. And what we do is we add some buffer salts together with the drink solution, so that the pH is well balanced, and we see that we can show in biodistribution studies that the modified bacteria survive the acid medium and arrive in the GI tract. So that has been shown in the clinic and in preclinical studies. Thanks for the question.
Great, thanks. All right, so I think we should probably wrap up. Maybe we could have the panelists go around and have some sort of final thoughts about this, where we are in sort of this inflection point in gene and cell therapies and novel modalities, and recommendations for next step in demonstrating your value. I think we're going to start seeing a lot more clinical results, positive and negative. We have some hazes of some safety issues that are emerging around some of the viral vectors out there, both lenti and AAV. Even though I think it's controversial if it is really implicated or not. So, as we start to advance these programs and get more clinical data, we're going to understand the real, benefit that they're providing clinically, durability-wise, and sort of unforeseen safety issues. So, any last recommendations that the investors have as, programs move forward in the clinic? Jay, do you want to start?
Yeah, From my perspective, it's very important that a company that pitches to us, with a new technology, new modality, needs to really have a good idea about the low-hanging fruits. How can they develop a product as fast as possible that is also able to compete in the environment, and not to forget about production.
I would agree with that. I think having an experienced management team with a track record of transparency and managing expectations and ability to build an organization and advance drug candidates, drive clinical progress, and collaborate with regulators, scale up manufacturing, and eventually bring some commercial experience to the table. All of those things are positive signals for investors.
Great. Sondra, any final comments?
Yeah, just to say, I agree with all that, and I think we don't have that much time, but one of the conclusions is these technologies work. So there's no doubt in my mind that we will see more success. And if I would put my money somewhere, for example, I would say the next step, solid tumors and CNS disease for gene therapy.
Great. Regan, any final comments, thoughts?
No, just in my field in TCRT, looking forward to some of our competitors' clinical data to sort of pave the way for the field, to be honest. I mean, like I say, very, very immature field, and we'll be contributing in clinic very soon, but there are some trailblazers that we are following, and we're looking forward to their clinical data to show TCRTs are effective, which we firmly believe they are.
Great. And Heinz, your final words.
Yes, thank you. Yeah, I think we are on a good track to generate some good data. We have some readouts, and one of the advantages of recurrent glioblastoma is that the time to read out is not very long, as everybody knows. And I think we have a good communication strategy, so I hope we can surprise you with some good data later this year.
Great. Well, thank you, everybody. We are out of time. We just sort of skim the tip of the iceberg, I think, with this conversation and, and the panelists that are here. But, more to come. And I hope that the audience took away some, good nuggets of, some advice in how to demonstrate and communicate the value of your platforms. And, investors got a good view into these two biotech companies. Thank you.
Thank you.
Thanks, everyone.
Thank you, everyone.
Thanks.